आईडी बताइए।
उनको पूछ रहे हैं कि पूछ लेते हैं कि सर उनको लेट होगा तो टेलीफोन में करते हैं। You can if you want, you can stay. Sir, shall we start with your address only?
Sir, you decide. I'm available here, and you had this opening remark, I guess,
Yes,
as
sir.
I understood from the agenda that you sent me across.
Yes, sir. So shall I go ahead with that first and then?
Please go ahead, sir.
Thank you, sir.
Yes, sir.
अब आ रहे हैं, आप लोग शुरू करो। अब आ जाओ। ना अभी खाली शुरू कर दीजिए डायरेक्टर साहब।
स्थापना दिवस का हार्दिक स्वागत और अभिनंदन। जैसे कि हमें अभी समाचार प्राप्त हुआ है कि हमारे चीफ गेस्ट कुछ ही पलों में हमारे बीच पधार सकते हैं, लेकिन टाइम कंस्ट्रेंट होने की वजह से अभी गेस्ट ऑफ ऑनर विशिष्ट अतिथि क्योंकि डॉक्टर समीरन पांडा, एडिशनल डीजी ICMR एंड हेड ECD ऑलरेडी हमारे साथ ज्वाइन कर चुके हैं वर्चुअली। इसके साथ ही प्रोफेसर एनी हेलक from Netherlands she has also joined virtually. So we can start with the address of the guest of honor. I'm so sorry. मैं स्वागत भाषण के लिए एड्रेस के लिए हमारे रिस्पेक्टेड डायरेक्टर सर डॉक्टर अपूर्व दास, डायरेक्टर NIRTH को स्टेज पर आमंत्रित करती हूँ कि वे स्थापना दिवस समारोह का शुभारंभ स्वागत भाषण देकर करें और जनजाति स्वास्थ्य में NIRTH द्वारा किए गए अनुसंधान कार्यों से हमें अवगत कराएं। So please.
Good evening, everyone. Since we are running short of time and our chief guest of this occasion will be arriving soon, but with the permission of the ICMR headquarters, Dr. Samiran Panda, who has already joined. So we'll start the proceeding, and then once the Honorable Vice Chancellor will arrive, then we will also start with it. So first of all, I welcome you from the ICMR-NITHR, Jabalpur, from all the staff, and I'm really thankful that Dr. Samiran Panda, who is representing the Director General of ICMR and Secretary DHR, Professor Balaram Bhargav. So he has joined already, and I'm officially welcoming him, and also I'm welcoming Professor Anani K. Halak, who has connected with us from in the Netherlands. So she will be delivering the scientific address for the Foundation Day. So first of all, I would like to show a few slides on the activities of ICMR-NITH, which we are conducting since the inception of this institute. And then also I will put forth that what are the plans and what we are doing. So, sir, as you see in this slide, we are an institute. It has been established in 1984 in two-room house in the adjoining medical college. And then the main focus was to cater to the national problem and health problems in tribal population, in common communicable diseases, environmental health problem, and mainly the hemoglobinopathies, which are blood genetic disorder. It was then named as the Regional Medical Research Center for Tribals, and it has shifted to the present campus in 1992, and then the offices and laboratory were also shifted accordingly. Sir, the main objective of this project was to plan, conduct, and coordinate research in order to bring out specific health problems and health needs of tribals of the country, to conduct epidemiological studies of communicable and non-communicable diseases among the tribals, to investigate hemoglobinopathies in tribal and other communities, and to advise and assist the government in planning, executing, monitoring, and evaluation of tribal health programs, and in training of health functionaries. So we have from Regional Medical Research Center in 1984, we have been renamed as National Institute of Research in Tribal Health in 2014. So since then we have traveled very far, and we have collaboration with all the international and national organizations as shown in the slide. Now we have a nationwide collaborative presence in at least 10 different states of India, where we are conducting in various diseases in tribes of this country. Next, please. So these are the main focus of this institute: communicable diseases, hemoglobinopathies, non-communicable diseases, viral and zoonotic diseases, and social science and ethnomedicine. Next, please. Sir, I'll show you some of the important research which we have taken up over the last 39 years. This is one of the projects which have been conducted very recently. It has been just finished. It was a PPP model to eliminate malaria in a tribal district of Madhya Pradesh. It consists of 1,233 villages, and with a consistent plan of surveillance, pest management, vector control, and training and capacity building, we have actually developed a model. In 36 months of operations, we have shown that malaria incidences can be reduced up to 90%. And if you see the picture below, the graph shows that this was the baseline data, and then in end line, malaria incidences have been actually decreased very drastically. Next, please. So this is a project which is a dream project of this institute. The ICMR headquarters, especially Professor Balaram Bhargav, who is the Secretary DHR and DG ICMR, he has entrusted us to show a model, to develop a model to decrease the prevalence of tuberculosis in a particularly vulnerable tribal group, which is called as Saharia. So since many years, we have been conducting research in this particular tribe for reduction of TB. And then if you see in the slide that prevalence of TB sometimes goes up to 3,000 in some districts, in those districts where Saharia is located. And with our research, we could reduce the prevalence to around 2,000 per lakh population. But this is still high because the national average is around 200. So this research we have already done. Around 70,000 TB cases have been screened in 7 districts. Around 9,000 cases are detected, and notification rate in those districts actually rose up about 4 times, and the success rate was more than 91%. So decrease in loss to follow-up and treatment failure was also have also been taken care of. Next, please. Recently, actually, we have inducted more innovative approach of having handheld X-ray devices in the field and on-spot molecular diagnosis, contact tracing, ready-to-use therapeutic food, and these are in addition to the programmatic approach of TB elimination, and we are doing it in collaboration with ICMR headquarters scientists, Central TB Division, Panchayat Raj Department, State TB Department, DHS of Madhya Pradesh, and Health and Wellness Centers. And we are developing a model in Shiopur district of Madhya Pradesh, where Saharia tribe is highly dominated. Next page. Talking about hemoglobinopathies, mainly we are concentrated to sickle cell anemia, and we have done screening. I'm actually proud to say that the map which is available for Madhya Pradesh in tribal areas for sickle cell disorders, this map is still a research work conducted by us only, and we have also conducted newborn screening around 8,000. We are conducting diagnosis and patient follow-up. For that, we have established a cohort in the nearby medical college. And then since last 4 or 5 years, we are also providing therapeutic intervention and follow-up and counseling of patients and their caregivers also. In terms of capacity building, we are constantly providing training of scientific and technical staff of Tribal Research Institute in 19 states, the TRIs, and also to the health officials in 19 states of India in collaboration with Ministry of Tribal Affairs, Government of India. So this is a very important activity. I would say that it's a milestone. How chlorosis can be prevented. So this prevention of chlorosis model has already been adopted by the national program. Our scientists have worked consistently. And then if you see the picture in 1995, this is the baseline data, a child with skeletal chlorosis, and then after intervention, how the current status of this child looks like. He has almost recovered from chlorosis. This is a model which has been developed by this institute. Talking about viral diseases, we are currently providing, we have a viral research and diagnostic laboratory in our institute. We are providing diagnosis of around 23 viral diseases using molecular and serological tools. And we also conduct serological, molecular, and epidemiological research. We have a biosafety level 2 in our institute. And we have also a rapid response team to investigate viral disease outbreaks in various viral diseases in this region. So regarding COVID-19 research and diagnosis, we have conducted 3 rounds or 4 rounds of serosurveillance in different places of Madhya Pradesh. So far, we have tested more than 200,000 samples for COVID-19. And also we have 3 different projects on COVID. One is psychological impact on COVID-19 pandemic, and one is understanding the perspective and coping strategy of COVID-19 in tribes. And then we have also sequenced the whole genome of COVID-19 in MP. So we have a project which is a networking project from ICMR regarding universal health coverage among scheduled tribes. For this, we are working in Shiopur district, which is highly dominated by Saharia tribes. With this different phase, we have 3 phases, and then goal is to provide universal health coverage extending to the tribes of India. This is a research project. This is a laboratory which has been set up very recently to do research in different mycobacterial diseases, specifically molecular drug susceptibility testing, PCR-based amplicon sequencing, multiplex PCR, LAMP assay, and development of field-friendly point-of-care diagnosis. So we are using the genome-wide analysis of microbial pathogens, for example, Mycobacterium tuberculosis, Mycobacterium leprae, SARS-CoV-19, COVID-2 virus, Oriensiasis susugamusi, which is a staph pathogen, and also HPV. So these are some of the 4 different projects which we want to be involved in the future. One is to looking at the high burden of sickle cell diseases in tribes, especially in Madhya Pradesh and in central India. We wish to develop or establish a center of excellence in sickle cell diseases. We also have taken a place to reduce high tuberculosis burden in Saharia tribe of Madhya Pradesh. As I already mentioned that we are involved in one district to develop a model. And once the model is developed, then it will be expanded to other 8 different districts so that we can at least reduce the tuberculosis burden in this Saharia tribe. And we have actually developed a model also for connecting the traditional tribal healers into public health. This is one of the projects which has been taken by ICMR headquarters, and the headquarters is very interested to fund this kind of project so that the tribal communities which are not using the public health now, they will be able to be connected. And then we are also interested to know, to understand the health and nutritional status of different, particularly vulnerable tribal groups, which are, there are 75 PVTGs in this country. So once we know, because we know that nutrition is the main key for disease manifestations. So we are also interested to have such kind of project to understand the health and nutritional status of the different tribes. Thank you so much for for patiently listening. And now I am handing over to Dr. Nisha Thakur, who will be carrying forward this program. Thank you very much.
Thank you very much, sir, for giving an overview of the research activities of ICMR-NITH. कार्यक्रम के अगले चरण में, जैसा कि आप सभी को विदित है कि आदरणीय अतिरिक्त महानिदेशक महोदय ICMR नई दिल्ली ने आज के समारोह में विशिष्ट अतिथि के रूप में वर्चुअली पधारकर हमें अनुग्रहित किया है। और वे किसी परिचय के मोहताज नहीं है, उनके बारे में कुछ कहना सूरज को दिया दिखाने की तरह है। फिर भी मैं कुछ अचीवमेंट्स उनके आप लोगों के साथ शेयर करना चाहती हूँ। डॉक्टर समीरन पांडा, एडिशनल डायरेक्टर जनरल ICMR, head ECD, is a tropical medicine specialist by profession. He has conducted infectious disease research in community settings. His remarkable work includes HIV drug use, conducted training in many South and Southeast Asian countries. He has edited several books and authored articles in peer-reviewed international journals. His recent exceptional contribution during COVID-19 pandemic is worth mentioning here. He has worked with UNESCO, UNODC, and WHO in several countries. With this brief introduction, I am honored to call upon our guest of honor, Dr. Panda, to address the August gathering and guide us. So please.
Good evening. Dr. Aparup Das, Director of the National Institute of Tribal Health, which is one of the very well-known permanent institutes of ICMR. The guest speaker and the scientist from the Netherlands who has joined us through virtual platform, Dr. Annemieke Gallup, and colleagues, scientists, researchers, friends. The institute, ICMR National Institute for Research on Tribal Health, has traveled a long path. And from a regional medical research institute, it has gained a status of the National Institute. So that's obvious. A very great achievement in terms of what the institute has been able to do and has been recognized for. However, at this juncture, I will share just a few thoughts. In the best interest of time, I will keep them brief. But the first thing that came to my mind while I was listening to Dr. Das, the director of the institute, that there have been things really well in the past and which are worth showcasing. However, what is the need of the hour? One is during COVID-19, it's a pandemic, it hit, every country globally. And same was the situation here in India. And ICMR institutes actually lived up to the occasion in terms of conducting not only research, but undertaking some of the interventions, informing the state health authorities. And that was the need of the hour. And the institute, ICMR-NITH, like any other ICMR institute, performed very well in terms of responding to the COVID-19 pandemic. So that was an acute phenomena, and that was responded to. But in addition to that, the institute, as very clearly presented by Dr. Das, was dealing and has been dealing with some of the acute infectious diseases and some of the non-communicable diseases. So clearly there are two areas. What do we need to do more, or do we need to look into ourselves or our work differently? That's the question. I remember reading a book quite some time ago on change, and I remember a statement there: any change is difficult, and I would be surprised if any change is suggested to the institute appears very difficult to the scientists and to the director, which holds true for me as well. For any human being, any change, any suggested change appears difficult because it requires coming out of the comfort zone and planning or looking out for something different, because that's the need of the hour. So are we introspecting? It is a question that is not specifically related to ICMR and IRTH, but it is relevant to all. But this is a question which is close to my heart. And as changes are difficult, we sometimes do not even ask this question: what is the change that is required, or that is the need of the hour? So I would say that it's high time we go to the tribal population that we cater to, or the institute is supposed to work for them. And well, working for them is fine, but are we working with them? So, and do we have the tribal population with us as our research collaborator? That's a fundamental question I'm asking. So it is probably not enough to working for them. It has to be by them, and it has to be with them. And in order to be able to do so, we need to ask questions: how explicitly we have ensured participation of the tribal population. It's not enough saying that, okay, in our project staff, so many people are from the tribal groups or population groups. So my fundamental question is, asking a research question even requires engaging with them. So that's my first point. My second point is about, if the change is necessary, how do we ensure going towards it? Very recently, I had to take part in the assessment process. Of course, there were external assessors for scientists of different categories: scientist B to C, or scientist C to D, or D to E, E to F, and the highest level of F to G. While I had to be present in all these assessments, I realized that things which are not being discussed, probably, I'm not specifically talking about NIRTH, I'm talking about ICMR institutes. We are all members of the same family, the big ICMR family. I believe that it is essential for us to discuss things like how to present the research findings and publish them. And can not that be Friday's seminar topic? Should it not be on a regular basis be discussed? And so that it is not after 3 years or 4 years or 5 years when a particular scientist is appearing for the assessment. Should it not happen consciously on a regular basis in terms of this particular skill, not only oratory skill or presentation skill through a PowerPoint, but how to present the data that we generate through the research and publish it? Because I won't say this whole adage, old one, which is publish or perish. I would rather say publish and flourish. Because the more we do a research, and the more we publish, we disseminate it widely, then somebody else expresses his or her willingness to collaborate. And so I am actually flourishing, I'm blooming, because Dr. Das presented in one of his PowerPoint slides a beautiful diagram. I really liked that. The network, how ICMR-NITH has been able to establish network with national and international institutes. My question is, how the junior scientists are able to establish their network with the fellow researchers. So institute is fine, but a researcher finally at the end of the day should be able to feel very satisfied, thinking that, okay, I conducted some research and out of interest, other researchers in other parts of the country or in other parts of the world reached out to me and tried to join hands or try to establish a dialogue. Is that happening? And if that happens, then an individual researcher also feels very satisfied and feels that he or she is growing. So that's my second point. The environment for growth. The scientists need to feel very satisfied being in the institute. So we need not to worry during the assessment after 3 years or 4 years or 5 years. If we feel satisfied, naturally, the passion with which we work, that would go. Of course, we won't be successful. We start with a question, we try to find answers to the questions, but not that we'll be able to find answers to all the questions that we asked. But at least we'll be able to find out answers to 2 or 3 of them. My third point is about socio-behavioral research. It's easier said than done. And particularly, we're very comfortable because if we have laboratories, if we have gadgets, if we have equipment, it's easy to generate data. So laboratory-based investigation structured questionnaire-based investigation, epidemiological or clinical in nature, most of us, I won't generalize, but most of us are trained on that. And we do not dare to venture in the field of qualitative research. But while quantitative research, mostly the observational studies, help us to cover a wide ground, knowing why is something happening if something is happening, and how is it happening if something is happening in terms of the health issues or in terms of the disease-related issues, why and how, in order to have those answers, in order to be able to go in depth, we need to undertake qualitative studies as well. And the toughest of all is the mixed method study. You have to justify why mixed method and in which sequence it should happen. So I would encourage all the scientists. I do not consider scientists as junior or senior, depending on their association with the institute. But I consider researchers as someone who is critically, asking or examining a particular question or a problem and is trying to find an answer to that. And there is a saying by Einstein that if I'm given a problem and I'm given also 1 hour time, I would rather, spend 50 to 55 minutes to try understand the nature of the problem, because finding out a solution then would be easy and 5 minutes should be good enough if I've understood the problem well. So understanding a problem, and how do we understand the problem? That brings me back to the first point that I was asking. So understanding a problem should happen with the tribal population and the groups with whom we are working. So it's not the way we understand. Yes, sickle cell disease we know, tuberculosis we know about some non-communicable diseases. Sickle cell disease is a genetic disorder. And then, but then what is bothering the community? And how do we even ask a question by engagement or through engagement of the communities is the key point, moot point that I'm trying to drive home. I wish you all the best. I thank you, first of all, and for giving me this opportunity so that I could share my thoughts. Under Dr. Das's able leadership, I'm sure that the Institute will flourish. But these 3 points that are raised are just for your consideration. Thank you once again. Thank you all, and wish you all the best.
Thank you also for your inspiring speech, your valuable suggestions and guidance in achieving the goal to provide better health to our tribal brothers and sisters. And it will really benefit us all, especially for the budding scientists. Thank you once again. Now moving ahead in the series of today's event, it's time for the Foundation Day lecture that will be delivered by Professor Annie McKee-Halleck. Professor Halleck is a professor by special appointment at Leiden University Medical Center, Leiden, the Netherlands. Professor Halleck is working in the area of leprosy and TB for several years and has conducted multi-country, multi-centric studies that have led to the development of field-friendly diagnostics for leprosy as well as for tuberculosis. The title of the talk is Field-Friendly Diagnostic Test for Leprosy and Tuberculosis. With this brief introduction, I would request Professor Halleck to deliver Foundation Day lecture. Professor Halleck.
Thank you very much. I hope you can all hear me. And I'd like to thank the organizers for the invitation to provide this presentation now. I will start sharing my screen. And please indicate whether you can see my slides now.
Yes.
I can see the, yes, you can? Okay, good. Yeah, so I'll go to presenter mode for that. So as indicated already, I'll try to provide you a brief summary of all the work we've done on field-friendly diagnostic tests for leprosy and tuberculosis. And I think I minimized, yeah. And so first, I want to introduce myself a little bit more. So I work at the Department of Infectious Diseases at the Leiden University Medical Center. And I do a lot of work abroad, as you can see here, so mainly in Bangladesh, but also in India, Brazil, Nepal. And I have met, several years ago, I've met Dr. Pushpendra Singh, and together we also were able to get some funding for Spark on the diagnostics of leprosy and TB. And so brief background on the department that I work at. So we focus a lot on mycobacterial research, so Mycobacterium leprae and Mycobacterium tuberculosis, but also others. And we work on the development of vaccines, of drugs, and do a lot of basic immunological and molecular biology research, and also finally a lot of work on diagnostic tests. Well, for those of you who are not quite familiar with leprosy, so leprosy is a neglected tropical disease caused by Mycobacterium leprae, and in most cases nowadays are still found in India, Brazil, Indonesia, Bangladesh, and several African countries, so as Ethiopia, Tanzania. So it primarily affects the skin and the nerves, and it ranks second after tuberculosis in the order of severe human mycobacterial diseases. However, it is not very contagious, even not in the areas where leprosy is endemic, and it can be treated efficiently with multi-drug therapy. However, there are still a lot of challenges faced in leprosy research. And the most important one is that there is a stable number of new case detection each year. And 10% of those new cases are also children. So this indicates that transmission of the mycobacterium is still ongoing. And therefore, it is very crucial to early diagnose leprosy and leprae infection, so that irreversible handicaps that can occur after Mycobacterium leprae infection can be prevented. Well, at this stage, there are no tools for early diagnosis of leprosy, and there's also no tools for prediction of leprosy. And the diagnosis, therefore, occurs mostly clinical. So that is assessment of insensitivity or of patches on the skin. So what is happening after a person encounters Mycobacterium leprae? So when it's transmitted to individuals. So most individuals will be able to kill Mycobacterium leprae. That is what is seen on the left side. So an estimated 99% of those individuals. However, in some individuals, Mycobacterium leprae can survive, and it usually takes about 2 to 6 years before a person then develops leprosy, if they develop leprosy, but it can also occur after 10 or even 20 years. So during that time, Mycobacterium leprae remains the individuals that are infected and that are asymptomatic, and they can also transmit the bacterium. So, and therefore, it is important that we identify those individuals that are asymptomatic but still carry Mycobacterium leprae. Well, and as you can see in this slide, what is occurring after Mycobacterium leprae infection also depends on the person's immune system or the person's genetics, if you will. And some individuals develop a very strong cellular immunity, depicted here in this slide on the left, and some individuals a very strong humoral immunity. So cellular immunity means Th1 T cell responses, whereas humoral immunity means a lot of production of IgM, IgG, and sometimes IgA. So the persons on the right with high numbers of bacteria also have high numbers of antibodies and are not able to kill bacteria. And that is the most infectious phase of the disease. Whereas on the left, if they have a lot of cellular immunity, they can kill bacteria, but sometimes this immune response is too strong so that the T cell response function as a kind of autoimmune disease, so destructing their own tissues. Another issue that is impeding the detection of Mycobacterium leprae infection and thus of transmission is the very long incubation time. And I wanted to show this slide with you because it's a very exemplary way of in which you can see the long duration of the incubation period. So this is a chimpanzee in a Japanese zoo that was captured as a young animal in Sierra Leone in Africa. And 30 years after it developed a leprosy, so multibacillary leprosy. And it had skin lesions and positive PGO1 and positive test for leprosy. So it was very clear that the infection did not occur in Japan in the zoo, but had to have occurred in Sierra Leone, where there is still Mycobacterium leprae transmission. So this is a good example of that it can take a very long time before leprosy becomes apparent. And in that time, the individuals that are infected can still transmit the bacteria. And this is also what we, for instance, see in the Netherlands. So in the Netherlands, we don't have a lot of leprosy cases, but we do have some cases. Usually, these are individuals that have either traveled abroad or were born abroad or their parents migrated. And after decades, they then can develop leprosy. So to summarize this part is, so what happens after Mycobacterium leprae is transmitted to an individual? Well, only a minority develops the disease. The way they become infectious is heterogeneous. Some do, some don't. And also the kinetics, so how long after exposure do they develop disease, is also different. And what kind of immune response, so either antibodies or T1 T cells, also differs a lot. So it's a heterogeneous disease. So we refer to it as the immunological spectrum of leprosy. There is still another issue that is complicating nowadays the transmission and the efforts to reduce the transmission. And that is that humans are not the only source of Mycobacterium leprae transmission. So they can also, Mycobacterium leprae can also be transmitted by armadillos. And there are recent reports on that in Brazil, but also now very recently in Mexico. And particularly those individuals who interact with raw meat or raw blood or kill or hunt armadillos can develop leprosy in, for instance, the southern states of the USA. And then in 2016, you may have heard that also another animal, which was the red squirrel in the British Isles, so Scotland or in south of England, were seen that had developed similar lesions and were found to be positive with Mycobacterium leprae or Mycobacterium lepromatosis. And this was work from Charlotte Lavancy and Anne Meredith and co-workers. So we also collaborated with that group, and together with also the researchers in Suriname, where there was a lot of armadillos, and collaborators in Bangladesh, where there's a lot of human leprosy patients. We collected soil samples from each of these parts and found that we could detect Mycobacterium leprae DNA also in soil. Of course, we couldn't say whether this was derived from still viable, living Mycobacterium leprae, but, sorry? Okay, but what we could see is that there was everywhere where there was either animal sources or in fact humans, that we could find it also in the soil. So this can also be a way of transmitting the mycobacterium. So to summarize, it's very crucial to diagnose early whether people are infected with Mycobacterium leprae so that we can intervene in that incubation part, so to reduce the transmission. Most of the transmission occurs via human-to-human interactions, but there are also either non-human primates or other animals that carry Mycobacterium leprae, and even in the soil it can occur. So in countries where elimination of leprosy is something that is well, either ongoing or focused at, we have to keep in mind that not only human interactions are important, but that also animals can play a role in this last step towards elimination of leprosy. So we're working towards diagnostic tests for this particular phase, so the Mycobacterium leprae infection without any symptoms, so that either individuals that are positive in such a diagnostic test can be treated with multi-drug therapy for disease, or with post-exposure prophylaxis methods, either immunoprophylaxis by BCG vaccination or treatment with the WHO endorsed prophylactic therapy, one single dose of rifampin. So why do we choose for that immunodiagnostic test and not, for instance, detection of bacteria? Well, that is because if pathogens like viruses or bacteria or helminths, if they interact with a host, then all kinds of cells start to get activated because the immune response wants to eliminate the pathogens. And during that elimination process, there are a lot of biomarkers that are produced by these cells, and they leave a kind of footprint of infection or of disease. And that is why we are interested in biomarkers for disease and infection. And usually what we do is we study endemic populations, and various cohorts are included, those that are not infected at all and are healthy, or those that are either infected or susceptible or have close contacts with people that have disease, and of course the clinically diagnosed leprosy patients. And we compare the biomarker profiles of those individuals. And for leprosy, in contrast to TB, the humoral response, so that means the antibody response against the bacteria, play an important role, because this includes, for instance, the antibodies directed against phenolic glycolipid 1, also called PGL1. And PGL1 is namely a major part of the cell wall of Mycobacterium leprae. It is a measure of virulence, actually, and antibodies against Mycobacterium leprae PGL1, they correlate with the number of bacteria. However, antibodies do not indicate disease, so they only indicate infection. And antibodies can also not discriminate between present and past infection. So, whereas we are lucky in leprosy research to have such a humoral response biomarker, it is still not really sufficient. And well, as I already mentioned, so there is this good correlation between the anti-PGL1 antibodies, as you can see here on the left. So if there is BI negative, so bacterial index negative individuals, they have low PGL1 levels, whereas BI positive high, and there is a correlation, positive correlation with qPCR, so the number of bacteria as estimated by qPCR lab, and the antibody levels, and also the multibacillary patients. And they have a lot of they have more new cases than individuals with, for instance, one lesion. So it is very clear that there is this relation between infectivity and antibody response, but it's still not sufficient to detect everybody that is infective. So what we need is biomarkers for post-bacillary disease and also for more early disease, because usually if you detect multibacillary patients, there is already advanced pathology and sometimes even irreversible damage. And also for active multibacillary disease, for some cases, for instance, in Bangladesh, we see MB cases that have a low BI. So there, in those cases, PGL1 antibodies is also not enough. So new biomarkers are required. And that is what we have been investigating, so the host biomarkers. So we identified them through omics approaches, being either transcriptomics, so the detection of RNA, proteomics, detection of proteins in host serum proteins mainly, or the detection of metabolites in serum or in urine. And just to give you a quick summary of years of research, we have recently identified a signature of 5 biomarkers that is specific for the whole leprosy spectrum. So not only the multibacillary, but also the post-bacillary cases and everything in between. And this 5 biomarker signature, if you combine all these markers, then it depends on the type of leprosy patients, whether a marker is able to identify, discriminate the individual from the contacts that are infected. But overall, the markers that are provided on the left are discriminating PB leprosy, whereas markers identified on the right, CRP, IP10, and the antibodies against PGL1, they are useful to identify multibacillary patients. So thus far about identification of the biomarkers for leprosy, but now what about translating that to the lab? Because of course we want to measure those biomarkers, not using any very complicated laboratory technique and apparatus. So we want to go to the field and be in any household, be able to use a test and directly near patient provide the results. So this translation from laboratory to field use, for that we use the so-called UCP lateral flow. And UCP stands for upconverting reporter particles. And this is schematic, this is shown on the right. There you see some examples of various colors that UCP can the lateral flow assay that we use is kind of field-friendly alternative for ELISA. So it's also based on a sandwich of 2 antibodies that recognize the same target. And what is important is that it's a quantitative assay. So that means that it's not like a plus or a minus results that we have in the field. No, we have a direct quantitative measure. So it means that there is a correlation between the signal that is measured with the test and the amount of the biomarker. So therefore, it makes it also very useful, for instance, to discriminate between the height of infection, but also to monitor any treatment. And but I will come back to that later. What is another advantage is that it's very sensitive, and that is because there is no background, because the UCP particles, they do not occur a lot in the regular environment. Finally, it's also very stable. So transport without a cold chain to any part of the world is not a problem. And finally, it doesn't bleach. So that means it can be read over and over again. So it can be read in the field, but then if you want to read it a day later or a month later or a year later, you can use the same strip again to have the same unbleached, undecreased signal. And that is because this is the upconversion process that causes that. So it's a luminescence process. So by using an infrared beam, which is in a reader that is used in the field, the structure of the UCP is excited, and then energy transfer can take place, which results in emission in the visible spectrum as luminescence, which is then detected as, for instance, 980 nanometers. And this you can, as I said, do over and over again with the strips. So here's, for example, several strips that we have been working on. I think this is an example of Brazil, and we can read the strips in Brazil at the endemic site, but we can also, take them later, years later, and read them over again. We get the same results. So this is very useful, particularly in the stages in which you are evaluating the test and want to measure it with a different point in time to check whether in the field the measurements have gone according to procedures. So since 2007, when we started working on the UCP lateral flow, we have incorporated various markers in our systems like interferon gamma, IP10, IO10, CCL4, ApoA1, CRP, etc. And we used it in serum, in fingerstick blood, in pleural effusions for TB, and also in whole blood assays. So that means that stimulated whole blood. And to give you an idea what it looks like, it's lateral flow like any lateral flow. So there is a little bit of blood on a sample pad, and then it will flow to the right. And it first encounters a test line where there is an antibody against a target of choice, and then a control line. Of course, this is only for a single biomarker, to detect a single biomarker. And then if there is a no target, then the test looks like this. So there's only a peak at flow control. But if there is target, and then there is a peak at the test line and the flow control line. I'm sure that any one of you has done a similar way of detecting whether he or she has antibodies against COVID, or whether she is antigen positive for COVID-19. And so this is the same principle. The only difference is that this is a quantitative assay, and it can be reread again, whereas most of the COVID-19 tests, SARS-CoV-2 tests, you have to discard them after 15 minutes. So some of the activities that we are performing now in the field, they will go as follows. So near patient, a finger prick sample of 20 microliter, which is taken with a Minifet to keep this quantitative aspect, because the Minifet is exactly taking up 20 microliters. This is provided through the cassette, and then we have a portable reader in which the cassette fits. It goes in here, and we get a quantitative result. So we get a quantitative result, so the test peak and the flow control peak. And this quantitative result is a measure of the amount of antibody present. Well, as you can envisage, measuring of one biomarker, although antibodies are very specific, one biomarker is simply not enough to get difference between those that have been heavily exposed, like contacts of patients, and those that develop the disease, like post-bacillary patients. So therefore, we worked on a lateral flow a multi-biomarker test, so-called MBT. And this multi-biomarker test can measure 6 biomarkers at the same time. So for instance, those 5 biomarkers that we identified, that I just showed you, that we identified for post-bacillary disease, plus, for instance, the antibody. So this is what we did, and we evaluated this in a high endemic area in Bangladesh, but also in a low endemic area in South Korea. And we could see here, this is on the right part, you see what the strip really looks like. So the flow direction is not from left to right, but in order to not interfere each line with the other markers, the flow direction is from the bottom to the top, so that each marker is encountered by the sample in the same way, and they don't disturb each other. So then we get an output with 12 markers, so a test and a flow control peak for each of the marker. And again, the ratio between the test and the flow control is a measure for each of the antibodies. So to summarize how we work on biomarker selection and implementation in multi-biomarker tests. So we first done a lot of proteomics, for instance, the Luminex or other techniques, which are laboratory dependent. And so the concentration and the differences between the test groups and whether the antibodies are available for detection of these markers, they all determine whether a marker can be continued to the next step, which is an ELISA, in which we test the antibodies for the markers and to see whether the concentration and the differences between the test groups are still sufficient, because we aim at a difference of 3-fold at least for non-disease versus disease. Well, then some of them had to be dropped, and we continued with several other markers to the UCP lateral flow, in which we determine whether the ratios, so the outcome of the lateral flow, are still different enough between the test group and the control groups. And finally, we look at the UCP multi-biomarker test and do the field evaluation with that. And then sometimes we find that some of the markers are not, are not compatible with each other, so we have to adapt that as well. And of course, what we need to do is, and that is something that is going on now, is we need to determine whether at each endemic site the same markers can detect the same test groups in the same way, and so that the multi-biomarker test can be applied globally. And this is a process that is ongoing now. So for your information, some applications, so for instance, home screening, what we did in Pará in Brazil, and but also screening in the lab in China and in Ethiopia. And of course, we have a lot of studies ongoing in Nilphamari in Bangladesh, where we screen in the villages for Mycobacterium leprae infection in context of leprosy patients. So to give you also a little bit information on what we're doing on TB, if time allows me to do so, what we do know of TB is that a lot of patients do not have access to diagnosis because they live in remote areas where for instance, the gene expert tests are not available or are too costly. So WHO estimates that there's around 10 million TB patients, and that a lot of them do not have, these do not have access to right TB diagnosis. And of course, that gap of 3.6 million is very crucial because it will impede the reduction of transmission of TB. And particularly this is, of course, now the case seeing the COVID-19 situation of the last 2 years. So what we are working on in several consortia together with African universities is a test for Triage Tb, and this point of care test should be a test that needs to be conducted in less than 1 hour, and then those individuals that are negative, which are usually a majority of the cases, they have other respiratory diseases, and those that are positive could have TB or not TB, but then there is a selection, and you don't need to send everybody to have an expensive X-ray or an expensive gene expert test. So that is the idea, to reduce the cost, to reduce transmission because of the ability to have early treatment. So we focus on a test like that, and in several projects, this type of TB test is being assessed. And here you can see that we started in 2010 and are now working towards the end to validate this Triage Tb test in several studies of EDCTP and NIH. And to give you an idea of the result that came from the analysis of the LUMC multi-biomarker test with markers for active TB that could discriminate active TB from other respiratory diseases. This is the result for Ethiopia, Gambia, Namibia, South Africa, and Uganda. The red dots indicate HIV positive individuals. And underneath each graph, you can see the area under the curve. No TB means the group of other respiratory diseases. So they did have complaints like weight loss, night sweats, cough, persistent cough. So, and these were, for instance, other pulmonary diseases, being cancer or pneumonias. And so the areas under the curve, they range from 0.83 to 0.89. So probably not enough yet for WHO, but really making progress in that. And this is all based on the number of positive markers in the multi-biomarker test that we have developed. And what I already mentioned is the quantitative aspect of the multi-biomarker test, which is important. And that means, for instance, that you can measure the differences. So if our test would have been a plus or minus test, then we wouldn't have been able to monitor treatment efficacy like you see here now. So from month 0 to month 2 to month 6 for the individuals under treatment, so the TB patient under treatment in these 5 African countries, you see a reduction in most individuals of the number of positive biomarkers as measured with a multi-biomarker test. So this means that the quantitative aspect also allows treatment monitoring. So finally, I would like to thank the individuals that have been collaborating with us. So Jan-Hendrik Richardus, also in the Netherlands at Erasmus University, but also University Federal Pará with Claudio Salgado, Moses Batista, Jan Spencer in Colorado State University, for the Bangladesh cohort who worked together with the Rural Health Program staff of Leprosy Mission, and in University in Edinburgh with Anna Karen and Anna Schilling on the red squirrels, and with Deanna Hagen in Nepal. The people from my group are shown on the left, and these are PhD students in my group or technicians in my group, and the only male individual there is the PI on the development of UCP lateral flow test, which is Paul Korstens. So I thank you for your attention. I hope you've enjoyed it and I hope it was clear. But if you have any questions, please let me know.
So, thank you, Remek, for this wonderful talk. It was really very informative as well as very inspiring that you are able to conduct these many trials in so many different countries which have so much differences in terms of their geographical as well as political systems as well as their scientific infrastructure. So just one question, if in the interest of time, I'm very much curious about what was the specificity for the postbacillary leprosy, or especially for the tuberculoid leprosy, compared to their likelihood of being subclinically infected with TB. So basically, I am asking how well you can differentiate between the subclinical PB leprosy versus subclinical TB. Yeah, thank you.
That's a very interesting question, Pushpendra. Thank you for that. So that is requiring a lot of discussion, I think, because when we measure the differences between the leprosy patients and the contacts, then we determine the area under the curve or the sensitivity specificity using the data of that same group. We have not yet been able to go to one area where there is both active TB cases and leprosy cases. But of course, you can envisage that if a person comes to a clinic with, skin lesions or loss of sensitivity, and not with persistent cough, then you would know that clinically this is probably a leprosy patient and not a TB patient. So you also must think about when are you going to use such a test, and also always include the clinic. So always include the way and where the patient comes to and what kind of complaints they have. So what use would it be to measure these two the differences? So you see my point?
Yes, thank you. Actually, in India, we do have some places where it would be endemic for both. We can discuss it later also. Thank you very much.
Yes,
all this.
That would be interesting. Thank you.
Contribution, dedication, and commitment to the mycobacterial infectious diseases. It was wonderful to have you with us on our special day. Thank you once again. जैसे कि आप सभी जानते हैं, आज के हमारे फाउंडेशन डे सेलिब्रेशन में कुछ लास्ट मोमेंट पर जो चेंजेस हुए हैं, तो अभी बहुत ही हर्ष के साथ मैं ये आप लोगों को कहना चाहती हूँ कि हमारे मुख्य अतिथि महोदय प्रोफेसर कपिल देव मिश्र कुलपति रानी दुर्गावती विश्वविद्यालय जबलपुर पधार चुके हैं। मैं उनसे अनुरोध करूंगी कि वे मंच पर आकर अपना स्थान ग्रहण करें और मंच को सुशोभित करें। आदरणीय निदेशक महोदय, आदरणीय डॉक्टर केपी साहा सर, वरिष्ठ वैज्ञानिक NIRTH जबलपुर, आप सभी का हार्दिक हार्दिक अभिनंदन और ICMR-NITH जबलपुर के 39वें स्थापना दिवस की शुभकामनाएं। वास्तव में NIRTH का स्थापना दिवस 1 मार्च को प्रत्येक वर्ष मनाया जाता है, परंतु इस वर्ष 1 मार्च को महाशिवरात्रि के उपलक्ष्य में अवकाश होने के कारण हम इसे इसकी पूर्व संध्या 28 फरवरी को मना रहे हैं। जैसा कि आप सभी जानते हैं कि भारतीय परंपरा में दीप प्रज्वलन का एक महत्वपूर्ण स्थान है। अपनी इस परंपरा को आगे बढ़ाते हुए मैं आदरणीय निदेशक महोदय से विनम्र निवेदन करती हूँ कि वे माननीय मुख्य अतिथि एवं हमारे वरिष्ठ वैज्ञानिक डॉक्टर साहा के साथ दीप प्रज्वलन करके आज के कार्यक्रम का शुभारंभ करें। सोमा सद्गमय तमसोमा ज्योतिर्गमय मृत्योर्मामृतंगमय। ओम शांति शांति शांति। अर्थात हे ईश्वर, हमको असत्य से सत्य की ओर ले चलो, अंधकार से प्रकाश की ओर ले चलो, मृत्यु से अमरता की ओर ले चलो। और इसके साथ ही हम ईश्वर से ये भी प्रार्थना करते हैं कि दीपक का यह प्रकाश अज्ञानता का अंधेरा दूर करे और हमें अच्छे स्वास्थ्य और समृद्धि का आशीर्वाद दे। आप सभी का बहुत बहुत धन्यवाद। और दीप प्रज्वलन के साथ ही आज के इस कार्यक्रम का शुभारंभ भी होता है। इस परंपरा में अतिथि देवो भव की परंपरा का निर्वहन करते हुए अब मैं कार्यक्रम के अगले चरण में हमारे आदरणीय निदेशक महोदय से अनुरोध करती हूँ कि वे हमारे मुख्य अतिथि का पुष्प गुच्छ देकर स्वागत करें, अभिनंदन करें। हम लोग क्या अर्पित आपको करें, भेंट लाए कुछ पुष्प। आपको बहुत बहुत धन्यवाद। Foundation Day of the Institute is the day from which the Institute has started its journey. It is therefore obvious to celebrate the day with due manner. It is also the day when the past, present, and future of the Institute come together and take pledge to uplift the Institute to the greatest possible heights and dimensions. While saying this, we should not miss thanking the founders of this great institution who had a vision of imparting such a high quality research in the area of tribal health. Without their vision and constant support and blessing, this institution would not have become what it is today. वैसे तो NIRTH के 39 वर्ष हो चुके हैं और जनजातीय स्वास्थ्य अनुसंधान की दिशा में अनेकों उल्लेखनीय कार्य भी कर चुके हैं। फिर भी मैं ये कहूंगी कि अभी तो असली मंजिल पाना बाकी है, अभी तो इरादों का इम्तिहान बाकी है, अभी तो तौली है मुट्ठी भर जमीन, अभी तो तौलना आसमान बाकी है। तुम सोच रहे हो बस बादल की उड़ानों तक, मेरी तो निगाहें हैं सूरज के ठिकानों तक। सर एक्चुअली आज हमारे यहाँ 2 सेलिब्रेशन हुए हैं। ऐसे ही होते हैं। मैं आप सभी से अनुरोध करती हूँ कि एक जोरदार करतल ध्वनि से हमारे मुख्य अतिथि का स्वागत करें। मैं डॉक्टर अलीशा ठाकुर, समस्त राष्ट्रीय जनजाति स्वास्थ्य अनुसंधान संस्थान NIRTH, भारतीय आयुर्विज्ञान अनुसंधान परिषद ICMR, स्वास्थ्य अनुसंधान विभाग DHR, स्वास्थ्य एवं परिवार कल्याण मंत्रालय, भारत सरकार, जबलपुर की ओर से माननीय मुख्य अतिथि का और यहाँ पर पधारे हुए गणमान्य अति विशिष्ट अतिथियों, वरिष्ठ वैज्ञानिकों, प्रशासनिक, तकनीकी एवं प्रोजेक्ट स्टाफ, शोध छात्रों, सेवानिवृत्त वैज्ञानिकों, अधिकारियों, कर्मचारियों, सपोर्ट स्टाफ का और वर्चुअल माध्यम से जुड़े हुए विभिन्न संस्थानों के प्रतिनिधियों और ICMR के अन्य संस्थानों के आदरणीय निदेशकों का हार्दिक हार्दिक स्वागत, वंदन और अभिनंदन करती हूँ। NIRTH ICMR का एकमात्र ऐसा संस्थान है जो जनजातीय स्वास्थ्य के लिए समर्पित है। आज का दिन हमारे संस्थान के लिए बहुत ही विशेष है क्योंकि आज हम सभी यहाँ पर अपने संस्थान का 39वां स्थापना दिवस मनाने के लिए एकत्रित हुए हैं और आपको ये जानकर बहुत ही खुशी भी होगी कि ICMR NIRTH आज जिस स्वरूप में है ये पहले ऐसा नहीं था। इसकी शुरुआत हमारे पड़ोसी मेडिकल कॉलेज नेताजी सुभाष चंद्र बोस मेडिकल कॉलेज में केवल 3 कमरों के साथ 1984 में हुई। इसके बाद 2002 में हम इस भवन में स्थानांतरित हुए और 2014 में इसे राष्ट्रीय संस्थान का दर्जा प्राप्त हुआ। अब मैं कार्यक्रम की को आगे बढ़ाते हुए अभिनंदन समारोह में के प्रथम चरण में आज श्री सुभाष गोडबोले जी, प्रिंसिपल टेक्निकल ऑफिसर जो सेवानिवृत्त हुए हैं और इन्होंने 36 वर्ष से भी अधिक समय तक अपनी सेवाएं संस्थान में दी हैं और वर्तमान में वे वायरोलॉजी विभाग में पदस्थ रहे हैं। ICMR की परंपरा का निर्वहन करते हुए मैं मुख्य अतिथि महोदय से निवेदन करती हूँ कि वे श्री गोडबोले जी का अभिनंदन करें। गोडबोले यहाँ पर उपस्थित नहीं है तो इस श्रृंखला में मैं आगे आदरणीय मुख्य अतिथि महोदय से निवेदन करूंगी कि वे NIRTH स्टाफ मेंबर्स उनका अभिनंदन करें जिन्होंने अपनी सेवाएं इस संस्थान को देते हुए 25 वर्ष पूर्ण कर लिए हैं। इसके लिए मैं मंच पर आमंत्रित करती हूँ श्री मदन सिंह मरावी, लैब असिस्टेंट 1, श्री प्रेम सिंह गौड़, लैब असिस्टेंट 1, श्री महेंद्र जयदेव यूके, टेक्निकल ऑफिसर B, थैंक यू वेरी मच सर। इस श्रृंखला में आगे बढ़ते हुए पोस्टल प्राइज डिस्ट्रीब्यूशन का समय आ चुका है and I'm glad to announce that recently on 8th of December 2021 His Excellency Honorable Governor of Madhya Pradesh Shri Mangubhai Patel visited our institute due to his keen interest towards improving the health of tribals. During his visit, a poster exhibition was organized to showcase our research activities, and later these posters were evaluated by a committee, and now we are distributing the best poster prizes to the winners, and there are 2 categories for first 5 best posters and second best 5 posters. So I will request our chief guest to please felicitate the winners. I will announce the names of the winners In the first category, first best 5 posters, Dr. Mukul Sharma, Research Associate. Akansha Kushram, Research Assistant. Ms. Nikhat Khan, SRF. Ms. Ravi Kanta Singh, Research Assistant. Mr. Anuj Mavlankar, Research Assistant. And the winners for the second best 5 posters are Ms. Poorna Dwivedi, SRF. Dr. Ashwini, Project Scientist 1. Dr. Subhasa Vishwal, Scientist B. Ms. Ruchika Sangle, Research Assistant. Ms. Nisha Chaudhary, MSW. Thank you very much, sir. Congratulations to all the winners. Felicitation के तृतीय चरण में मैं श्री एच.एस. ठाकुर हिंदी विभाग से अनुरोध करती हूँ कि वे मंच पर आकर ICMR हेडक्वार्टर न्यू दिल्ली द्वारा आयोजित हिंदी पखवाड़ा प्रतियोगिताओं के विजेताओं के नाम की घोषणा करें। और
नमस्कार। आप सभी को संस्थान के 39वें स्थापना दिवस फाउंडेशन दिवस की बहुत बहुत शुभकामनाएं। साथ ही हमने राष्ट्रीय विज्ञान दिवस भी मनाया, उसकी भी बहुत बहुत शुभकामनाएं। जैसा कि हम सभी जानते हैं, भारत सरकार की जो राजभाषा नीति है उसके तहत प्रतिवर्ष भारत सरकार के विभिन्न मंत्रालयों, विभागों और कार्यालयों में हिंदी दिवस, हिंदी सप्ताह मनाया जाता है, हिंदी पखवाड़ा, हिंदी मास मनाया जाता है। और ICMR या भारतीय आयुर्विज्ञान अनुसंधान परिषद के मुख्यालय में भी और उसके जितने भी देश भर में फैले विभिन्न केंद्र और संस्थान हैं, उनमें भी हिंदी पखवाड़ा और हिंदी दिवस बड़े धूमधाम से मनाते रहे हैं। आप सभी जानते हैं कि संस्थान में विगत वर्षों में हिंदी दिवस के अवसर पर और हिंदी पखवाड़े के अंतर्गत हम अपने संस्थान के वैज्ञानिकों, अधिकारियों और स्टाफ के लिए हिंदी की विभिन्न प्रतियोगिताएं आयोजित करते रहे हैं। आप सब लोग बड़े उत्साह के साथ भाग लेते रहे हैं। कोविड के कारण थोड़ा सा इस आयोजन का स्वरूप सीमित करना पड़ा इस वर्ष में, लेकिन फिर भी हमने पिछले 2 वर्षों में इस गतिविधि को जारी रखा और संस्थान में हिंदी पखवाड़े का आयोजन किया, हिंदी की विभिन्न प्रतियोगिताएं आयोजित की। भारतीय आयुर्विज्ञान अनुसंधान परिषद, जो हमारा मुख्यालय है नई दिल्ली में, पहली बार उन्होंने कोविड के कारण प्रतियोगिताओं का आयोजन वर्चुअली या ऑनलाइन किया। तो मैं समझता हूँ कि कोविड के और तमाम बुरे पहलुओं के साथ साथ ही एक उजला पहलू मुझे समझ में आया, जिसमें कि हम दूर दराज के क्षेत्रों से या विदेशों में भी बैठे, जैसे कि आज आप सुन रहे थे आज के हमारे स्थापना दिवस व्याख्यान के लिए नीदरलैंड से मैम हमें संबोधित कर रही थी, व्याख्यान दे रही थी और हम जानकारी उन्होंने लेप्रोसी के बारे में। इसी तरह से परिषद मुख्यालय द्वारा पहली बार जो हिंदी पखवाड़ा मनाया गया दिसंबर 21 को मनाया, और उसके अंतर्गत न केवल मुख्यालय के अधिकारियों कर्मचारियों के लिए, बल्कि पहली बार उन्होंने परिषद के विभिन्न संस्थानों में जो अधिकारी कर्मचारी वैज्ञानिक कार्यरत हैं उनके लिए भी उसमें शामिल किया। और बड़ी ही खुशी की बात है कि हमारे संस्थान के लोगों ने उसमें भाग लिया और सफलता के नए कीर्तिमान स्थापित किए, जो कि आप अभी थोड़ी देर में आदरणीय मुख्य अतिथि के जी के हाथों से जो पुरस्कार वितरित होंगे उससे आप समझ जाएंगे। तो उसमें उन्होंने मुख्यालय के लिए अलग प्रतियोगिताएं आयोजित की और संस्थानों केंद्रों के लिए अलग प्रतियोगिताएं आयोजित की थी। जो संस्थानों केंद्रों के लिए प्रतियोगिताएं रखी गई उसमें मुख्य रूप से 2 ही प्रतियोगिताएं थी, हिंदी वाक प्रतियोगिता या भाषा प्रतियोगिता और हिंदी कविता पाठ प्रतियोगिता। इन दोनों ही प्रतियोगिताओं में हमारे संस्थान के जिन लोगों ने भाग लिया उन्होंने पुरस्कार भी प्राप्त किए। ये हमारे लिए बड़ी खुशी की बात है। और अब आज ये एक बहुत ही बेहतरीन समय था, अवसर था हमारे सामने कि जब हम अपने संस्थान का 39वां स्थापना दिवस मना रहे थे। इस अवसर पर आदरणीय निदेशक महोदय का निर्देश था कि हम आदरणीय मुख्य अतिथि के हाथों से इन विजेताओं के लिए पुरस्कृत करें, उनको प्रमाण पत्र दिए जाएं, तो बहुत अच्छा रहे। तो निश्चित रूप से वह गौरवपूर्ण क्षण आ पहुंचा है और मैं आपके सामने जैसे कि निर्देश हुआ है इन दोनों प्रतियोगिताओं के लिए हमारे संस्थान के जिन कर्मियों ने पुरस्कार प्राप्त किए हैं मैं उनके नाम आपके सामने रख रहा हूँ। आदरणीय मुख्य अतिथि महोदय से सर से मैं अनुरोध करूंगा कि वो कृपया उनके लिए उनके जो प्रमाण पत्र हमें आदरणीय महानिदेशक महोदय के हाथों से हस्ताक्षरित प्राप्त हुए हैं वो उनकी विजेताओं को वितरित करने की कृपा। हिंदी वाक प्रतियोगिता, इसमें 5,000 रुपए का प्रथम पुरस्कार था और वह प्राप्त किया था श्री कौशल कुमार शुक्ल जी। कारणों से उपस्थित नहीं हो पाए हैं। और 1,000 रुपए का सांत्वना पुरस्कार डॉक्टर निशा ठाकुर जी ने प्राप्त किया था। मैं आमंत्रित करूंगा। दूसरी महत्वपूर्ण प्रतियोगिता थी हिंदी कविता पाठ प्रतियोगिता, जिसमें देश भर के विभिन्न संस्थानों केंद्रों से लोगों ने भाग लिया था हमारे साथियों ने। और इसमें बहुत ही गौरवान्वित करते हुए डॉक्टर निशा ठाकुर जी ने ही द्वितीय पुरस्कार 3,000 रुपए का प्राप्त किया था। और हिंदी कविता पाठ में ही तृतीय पुरस्कार 2,000 रुपए का प्राप्त किया था श्री सुरेंद्र सिंह। मैं उन्हें आमंत्रित करूंगा कि वो आदरणीय मुख्यातिथि महोदय से अपना प्रमाण पत्र प्राप्त करें। बहुत बहुत धन्यवाद सर। आदरणीय मुख्यातिथि महोदय का, आदरणीय निदेशक महोदय का, आप सभी का बहुत बहुत धन्यवाद जो आपने हमारे विजेताओं का प्रसार वर्धन किया, प्रोत्साहित किया। और इसी के साथ मैं अपना स्थान ग्रहण करता हूँ। आदरणीय डॉक्टर निशा ठाकुर जी आगे कार्यक्रम संचालित करेंगे। लेकिन पुनः मैं आप सभी को स्थापना दिवस की बहुत बहुत शुभकामनाएं देते हुए और अपने हिंदी कार्यक्रमों के लिए इन प्रतियोगिताओं में भाग लेने वाले सभी विजेताओं को बहुत बहुत शुभकामनाएं देते हुए वाणी को विराम देता हूँ। बहुत धन्यवाद।
बहुत बहुत धन्यवाद सर। गोदवले सर क्या हमारे बीच आ चुके हैं? यदि नहीं तो फिर हम कार्यक्रम की अगली श्रृंखला में बढ़ते हुए हमारे माननीय मुख्य अतिथि महोदय प्रोफेसर कपिल देव मिश्र, कुलपति रानी दुर्गावती विश्वविद्यालय का आभार व्यक्त करते हैं कि उन्होंने हमारे आमंत्रण को स्वीकार किया और अपनी व्यस्तता के बावजूद उन्होंने यहाँ पर आने का कष्ट किया और हमें पधार कर अनुग्रहित किया। Professor Kapil Dev Mishra is a renowned academician with more than 25 years of teaching experience. We feel privileged to have him here. He is the recipient of many national and international awards. Now, I am honored to call upon our chief guest for today's auspicious event, Professor Kapil Dev Mishra, Honorable Vice Chancellor Rani Durgavati Vishwavidyalaya, Jabalpur, to address the assembly.
बहुत बहुत धन्यवाद निशा ठाकुर जी। मंच पर विराजमान ICMR नेशनल इंस्टीट्यूट ऑफ रिसर्च इन ट्राइबल हेल्थ जबलपुर के डायरेक्टर डॉक्टर अप्रूप दास जी, जिनके नेतृत्व में संस्थान अद्वितीय कार्य कर रहा है। सम्माननीय डॉक्टर कल्याण बी शाह जी हमारे समक्ष बड़े सीरियस सब बैठे हुए हैं, प्रथम पंक्ति से लेकर और अंतिम तक। जैसे लगता है कि डायरेक्टर साहब ने कुछ कह दिया था, मुस्कुराइएगा मत। ऐसे हमारे सम्मानीय सभी साइंटिस्ट, वैज्ञानिक और शोध छात्र छात्राएं, फेलोशिप वाले भी हैं सभी। अरे भाई मैं भी देख मास्क लगाता हूँ, ऐसा नहीं है। इसी देश का वासी हूँ, जिस देश में गंगा बहती है, तो मैं जानता हूँ कौन मुस्कुरा रहा है कौन नहीं। और मैं विद्यार्थी भी हूँ, शिक्षक भी हूँ, टीचर भी हूँ और कुलपति तो हूँ ही। और हर रूप में इसलिए जानता हूँ कि अंतःकार में क्या चल रहा है। समय देख रहे हैं, घड़ी देख रहे हैं, घर की तरफ चेहरे से मालूम हो जाता है सुबह से चल रहा है कार्यक्रम, तो निश्चित रूप से थकावट होगी। और मैं बहुत बहुत बधाई देता हूँ 39वें स्थापना दिवस पर आप सभी को और संस्थान को। अभी मैं लेक्चर सुन रहा था, बहुत ही उत्कृष्ट कोट के, बड़े ही लाभप्रद। आप वैज्ञानिक हैं तो आप लोग उसके ज्यादा गहराई तक समझ रहे होंगे। मैं तो थोड़ा थोड़ा पकड़ पा रहा था सामान्य रूप से। और बहुत ही, जब मैं आया तो डायरेक्टर साहब के लेक्चर चल रहे थे। संस्थान के द्वारा जैसा बताया गया, बहुत ही महत्वपूर्ण गतिविधियां 39 वर्षों में प्रस्तुत की गई। और राष्ट्र की जो आकांक्षा है ट्राइबल्स एरिया में स्वास्थ्य के संवर्धन और उनके उत्कर्ष पर ले जाने की, उस आकांक्षा की परिपूर्ति यह संस्थान कर रहा है। हम सब आज बता रहे थे, डॉक्टर साहब की राष्ट्रीय विज्ञान दिवस भी मनाया गया। अमरकंटक के वाइस चांसलर हमारे मित्र प्रोफेसर प्रकाश मणि त्रिपाठी जी को जोड़ा गया था ऑनलाइन। आप सबको पता है कि स्थापना दिवस
अपने स्व को देखने का ही एक अवसर देता है। हमने जो बीता है, जो बीत गए वर्ष हैं, उसमें क्या किया है, वर्तमान में हमारी क्या स्थिति और फ्यूचर हमारा भविष्य क्या होगा। आप सबने देखा कि इस कोरोना काल में अगर किसी की निगाह थी कहीं, तो आपके इस अनुसंधान केंद्र की तरफ पूरा जबलपुर या मध्य प्रदेश की निगाह आप जैसे वैज्ञानिकों पर ही, और राष्ट्र पर जब जब आई क्राइसिस मैनेजमेंट कहीं हुआ है, तो ऐसे ही संस्थानों ने आगे बढ़ करके अगुवाई की है और समाज को राहत दी है। आज आप 39वां वर्ष मना रहे हैं, 11 वर्ष बाद 50 वर्ष आपका पूरा हो, 2033 में आप 50 वर्ष के हो जाएंगे और आप स्वर्ण जयंती मनाएंगे। बहुत से लोग इसमें, जितने युवा हमारे साथी बैठे हुए हैं, उन्हीं का दिन होगा, न हम रहेंगे न डायरेक्टर साहब रहेंगे, साहब भी इनका क्या रास्ता बहुत हंसी लग रही है, सही हास्य हंसने की बात। लेकिन 11 वर्ष के बाद तो हम स्वर्ण जयंती मनाएंगे, गोल्डन। तो देश और समाज और हमारा जबलपुर और मध्य प्रदेश जो सोच रहा है, आप सब जानते हैं कि हम सब इस देश के आजादी के 75वें वर्ष को भी मना रहे हैं, आप सब लोग भी मना रहे हैं, और सबको पता है कि राष्ट्रीय शिक्षा नीति संस्थाओं में प्रवेश कर चुकी है। ऐसे में बड़ा अच्छा अवसर है कि हम समाज के प्रत्येक वर्ग को, आप तो ट्राइबल्स में ही काम कर रहे हैं और भी क्षेत्रों में हमारे बता रहे थे कि इसमें MSW के भी स्टूडेंट्स हैं, तो समाज एक ऐसा समाज विज्ञान, सोशल साइंस, एक ही जगह साइंस जुड़ी है, वो सोशल साइंस में, और किसी में आर्ट्स के विषय में नहीं, ह्यूमैनिटीज में नहीं, साइंस उसमें जुड़ा हुआ है। 21वीं शताब्दी जिस तरफ जा रही है, उसमें टेक्नोलॉजी में हम लोगों ने महसूस किया कि सब चीजें हमारे हाथ में, मुट्ठी में। कोरोना भी वैसे ही है। आप सब जानते हैं कि सनातन काल से ही 2 प्रकार की प्रवृत्तियां चलती रही हैं, एक आसुरी प्रवृत्ति जिसको कहते हैं निगेटिव, और एक दैवीय प्रवृत्ति पॉजिटिव की। ये सदियों चलती रही और आगे भी चलती रहेंगी। एक तरफ कोरोना जैसे लोग कहते हैं कि कहां से उपजा है, किधर, उसका नाम मैं नहीं लूंगा, संकेत आप सब समझ रहे होंगे, कहीं आया है, ये लैब से ही तो पैदा हुआ था। अब बहुत प्रमाणिक क्या है, लेकिन आया किसी रिसर्चर के द्वारा ही दिया गया, लेकिन उसका निदान भी किसी शोध आप जैसे वैज्ञानिकों के माध्यम से ही दिया गया। तो हम सबकी संकल्पना है जो 21वीं शताब्दी की, और विशेषकर जो राष्ट्रीय शिक्षा नीति लेकर के आई है, और विश्वविद्यालय, महाविद्यालय, संस्थाओं में हम लोग जो कार्य कर रहे हैं, विशेषकर युवाओं के लिए, कि समाज का प्रत्येक वर्ग, चाहे जिस जात का हो, चाहे जिस धर्म का हो, चाहे जिस क्षेत्र का हो, चाहे जिस वर्ग का हो, क्योंकि हम सब जानते हैं प्रतिभा किसी की बंधक नहीं होती, न पंडित जी की होती है, न दूसरे जो ठाकुर साहब की हम लोग पूजा करते हैं, किसी की बंधक। अगर बंधक होती तो बाबा भीमराव अम्बेडकर को हम विश्व विजेता न मानते, और जो भारत के संविधान निर्माता करते हैं। तो इस युग में युगानुकूल जो परिवर्तन हो रहा है बहुत तेज गति से, वो ये है कि इन प्रतिभाओं को सुरक्षित, संरक्षित और पोषित करने का जो दायित्व है, वो नेतृत्व करने वाले का है, चाहे डायरेक्टर हो, चाहे कुलपति हो, चाहे आप जैसे साइंटिस्ट, और समाज का प्रत्येक वर्ग जिसकी चर्चा हम कर रहे हैं, कोई बीमार ना हो। कैसे ये संभव है? और जो बीमार हो कैसे स्वस्थ हो जाए? ये रिसर्च, ये शोध अपने को करना है। सर्वजन हिताय सर्वजन सुखाय, ऐसा शोध, ऐसा अनुसंधान नहीं। बहुत से अनुसंधान ऐसे आते हैं, हमको 7 वर्ष हो गए यहाँ पर, 7वां चल रहा है, बहुत हो गए, 1 टन 4 साल का होता है, एक ही पर्याप्त होता है। लेकिन किन्हीं कारणों बस कुछ बात होगी, इसलिए रोक दिया गया होगा, या प्रकृति का निर्णय। लेकिन शोध में ऐसी दिशा हो, आप सबसे मैं अपेक्षा करता हूँ, अपने आप ही अपने बच्चों से बात करता हूँ, अनुसंधानकर्ताओं से मैं बात करता हूँ, कि जो सबके हित के लिए हो, केवल वो लाइब्रेरी की शोभा बढ़ाने वाला ना हो, केवल टेंशन देने वाला नहीं, मुस्कुराने वाला। शिक्षा कैसी? जो मुस्कुराहट, जो दुखी मन को प्रसन्न कर दे, वो शिक्षा है, कदापि नहीं हो सकती। शिक्षा वो है जो बीमार को स्वस्थ बना दे, बेरोजगार को रोजगार युक्त कर दे, प्रदूषण युक्त को प्रदूषण मुक्त कर दे, और भ्रष्टाचार युक्त को भ्रष्टाचार मुक्त कर दे, जो विषमता को समानता में बदल दे, वह शिक्षा है। तो ये हम चाहते हैं, और इसी की तरह हम लोग काम कर रहे हैं। काम जरूर थोड़ा कठिन है, लेकिन युवाओं की जो टोली है हमारे यहाँ भी, आपके यहाँ, ये युवा मन बहुत तेज होता है, क्योंकि स्वामी विवेकानंद जी ने कहा है, सब जानते हैं कि युवाओं के प्रेरणा स्रोत स्वामी विवेकानंद जी हैं। उन्होंने कहा है कि इस राष्ट्र की संपूर्ण जो समस्याएं जितने भी प्रकार की, यदि कोई उनका समाधान उन चुनौतियों का देने में सक्षम है तो युवा ही है, जो आप लोग हैं। आप ये मत मानिएगा कि हमारे डायरेक्टर साहब युवा नहीं है, डायरेक्टर साहब भी युवा है। मन के हारे हार है, मन के जीते जी। और सच में डॉक्टर साहब से हमने कहा मुस्कुराए क्यों नहीं गए, वैसे सब चिंतन कर रहे हैं। और सच में जिस सभा में हम तो दिन भर काम हम लोग भी करते हैं, लेकिन अगर आप मुस्कुराएंगे नहीं, तनाव में ही रहेंगे, तो आपका शरीर, जो शरीर मन बुद्धि आत्मा है, फिजिकल मेंटल सोशल सोल, और जितनी चारों चीजें हैं वो कैसे प्रफुल्लित होंगी। और आप जानते हैं कि उसका प्रभाव मातृशक्ति के विशेष करके, चाहे वो पुरुष हो चाहे महिला हो, उस बच्चे पर पड़ता है, जो आने वाली पीढ़ी। अब रिसर्च सब हो चुके हैं, अब मनु की घटना सब लोग जानते हैं। अमेरिका ने बहुत पहले ये प्रमाणित किया था, कुछ वर्ष पहले आया था कि जब 4 महीने जब गर्भस्थ शिशु होता है, जो माँ सोचती है या उसके पूर्व पिता, वैसा ही बालक बालिका पैदा होती है। शोध से प्रमाणित हो चुका है। इसलिए आपका चिंतन, आपका मनन, आपका प्रफुल्लित होना, चाहे जितने बड़े आपको भगवान करे बड़ी ऊंचाई पर आप जाए, ईश्वर की तरफ से मैं कामना करता हूं कि ऐसा अनुसंधान करे जो विश्व प्रसिद्ध हो। लेकिन मुस्कुराना न भूलिए साथी, यदि आप मुस्कुराना हो जाएंगे तो सब शिक्षा किस काम? आपका बच्चा हो, आपका साथी हो, आपकी बहन हो, आपका भाई हो, हम लोग जो अपने यहाँ पति परमेश्वर, जो आप पति भी किसी को होंगे तो पत्नी ये चाहती होगी कि जब आए हमारे पतिदेव या हमारे भैया या हमारी बहन दीदी आ रहे हैं तो मुस्कुरा के मिले। क्यों चाहते कि नहीं? कोई चाहता है कि दुखी मन से मिले? कदापि। शिक्षा का उद्देश्य भी ये होना चाहिए हर संस्थान में, चाहे वो रिसर्च का हो, चाहे वो टीचिंग का हो, चाहे वो एक्सटेंशन का हो, और चाहे वो ट्रेनिंग का हो। यही 4 आयाम है, चाहे रूरल एरिया हो, चाहे वो नगरीय अर्बन क्षेत्र हो। पूरे परिवर्तन की दिशा यही चारों आयामों के द्वारा हम लोग परिवर्तित करते हैं। इंटीग्रेटेड अनुसंधान आपका विषय क्षेत्र है, और शिक्षण कार्य भी होता है। डॉक्टर साहब बता रहे थे कि हमारे क्लासेस भी चलती हैं कुछ ना, तो आज आवश्यकता बहुत है इस बात की कि आप प्रफुल्लित रहें। जिसके जेब में पैसा हो गए वही तो दूसरे की मदद करता है। बोलिए कि कर देते हैं बिना पैसे, आपकी जेब में नहीं कर पाएंगे, बोलिए, मैं पूछता हूं, नहीं कर सकते। तो जब आप प्रफुल्लित रहेंगे, खुश रहेंगे, तभी तो खुशी देंगे, दूसरे प्रफुल्लित होगा। इसलिए पहला काम, हमारे जितने भी विद्यार्थी, हमारे पास तो 2.5-3 लाख विद्यार्थी हैं, हजारों के ऊपर तो एम्प्लॉय है। हम उनसे यही कहते हैं, क्योंकि कौन ऐसा व्यक्ति है जिसको दुख नहीं है? कौन ऐसा? कुलपति जी से पूछिए, बैठिए, हम और डायरेक्टर साहब तो आप अकेले में सुने तो हम कुछ व्यक्त कर रहे हैं, डायरेक्टर साहब कुछ कह रहे। दुख किसको नहीं? प्रातः काल से शुरू हो जाता है। तो कहते नहीं, बहुत से लोग केवल अपने दुखों का गीत गाते हैं। बहुत से लोग केवल अपने दुखों का गीत गाते हैं कि होली हो या दिवाली सदा मातम मनाते हैं, मगर दुनिया उन्हीं की रागनी पर झूमते देखा जो जलती चिता पर बैठकर बांसुरी बजाते हैं, जो जलती चिता पर बैठकर। स्थापना दिवस पर मैं आप सबको बहुत बहुत शुभकामनाएं देता हूँ, क्योंकि आप में वही जो आत्मा जो डायरेक्टर साहब में है, मुझ में है, और हमारे साहा साहब में है, वही सब में है। चाहे हमारी मातृशक्ति हो और चाहे हमारे भाई लोग, सब में वही। आप अपने असीम शक्ति का प्रयोग करके क्या नहीं प्राप्त कर सकते? जो कहते हैं लोग आसमान में छेद कर सकते हैं, वो भी संभव है आप करने में सक्षम, और भारत के लोग कर ही रहे। विश्व में यूक्रेन ने किसी से सहायता की अपेक्षा की तो भारत से की कि आप युद्ध रोक सकते हैं। इसका अभिप्राय है कि हम हर क्षेत्र में आगे बढ़े। और यह शताब्दी 21वीं शताब्दी निश्चित रूप से जिस तरह वैक्सीनेशन को आपने कंट्रोल किया, जिस तरह से कोरोना को आपने कंट्रोल किया, जिस प्रकार से आपने विश्व में सहायता सहयोग किया, हर दृष्टि से आप आगे बढ़ रहे। मुझे लगता है युवाओं की जो टीम है वो आगे तो बढ़कर रहेगी, लेकिन प्रफुल्लता, आनंद, शिक्षा यानी जो आनंद नहीं देता है, आप कोई पिक्चर देखने जाते हैं, अगर वहां आनंद आपको सुख नहीं मिल रहा, छोड़ चले आते, पैसे की परवाह नहीं करते। तो आनंद से करिए, रिसर्च भी करिए तो आनंद से करिए, बड़ा आनंददायक रहेगा। और यही होता है सनातन काल के जितने भी आप अध्ययन करेंगे, हमारे जो अब तो धीरे धीरे सारी बातें प्रमाणित हो रही, हमारे यहाँ जो बातें कही गई हैं, पहले विदेश से वापस आता था तब कहा जाता था नहीं भारत में है, अब ये कहा जा रहा नहीं भारत में जो है वो प्रमाणित है। इसलिए पहले प्रसन्न होकर के, खुश होकर के आप शोध के, अनुसंधान के उस आयाम को प्रमाणित करें जो विश्व स्तर पर अभी चर्चा हो रही थी, तभी बात किया जा रहा। और आपसे बड़ी अपेक्षाएं हैं। ये संस्थान 39 वर्ष का हो गया, 39 वर्ष की उम्र बहुत होती है, बहुत सी विशिष्ट उपलब्धियां हुई। हमारे साथी लोग, हिंदी में प्रतियोगिता हुई और हिंदी में स्थान भी प्राप्त किया तमाम प्रतियोगिताओं में हमारे साथी लोग, मैं उनको बहुत बहुत बधाई देता हूँ। हमारे आपके संस्थान के एक आज सेवानिवृत्त भी हुए डायरेक्टर साहब, क्या नाम था, गोडबोले जी, गोडबोले जी, उनको भी बहुत बहुत शुभकामना देता हूँ। ये तो नियम ही है, हम भी 4 वर्ष के बाद हमारा भी सेवा समाप्त होगी तो हम भी 65 पर जाएगा, जहाँ जिस संस्थान में चले जाए, ये तो प्रकृति का नियम। और यहाँ से भी जाना है, आया है सो जाएगा राजा। इसलिए मस्त होकर के काम करिए। ये इसलिए मैं बता रहा हूं क्योंकि मैं शिक्षक बहुत नीचे से ऊपर आया। मैं भी कभी जो बैठे हमारा शोध सहयोगी था, आर्य था, JRF SRF हमको भी मिलती थी, लेकिन आज देश में जहां भी जाए तो आपको प्रथम स्थान मिलने का वो ये है कि प्रसन्न होकर के हर कार्य करिए। यही मैं अपने बच्चों को बताता हूँ, ये विद्यार्थियों को बताता हूँ, यही आप साथियों को मैं बता रहा हूँ कि खुश रहिए। और खुश रहने के दूसरे के पास कोई दवा नहीं है, अपने ही पास है, अपने पास। तो मैं चाहूंगा कि डायरेक्टर साहब तो कह रहे थे चिंतन कर रहे हैं, चिंतन करिए, खूब चिंतन करिए, मनन करिए, निध्यासन करिए और उस लक्ष्य को प्राप्त करिए जो संस्थान के द्वारा निर्धारित किया गया है, लेकिन आनंदमय रहिए। अभी हमारे और ICMR का डायरेक्टर साहब के नेतृत्व में और साथियों के नेतृत्व में बैठे हुए एक MU हुआ, सिकल सेल एनीमिया के उन्मूलन के लिए मंडला में 5 गांव हम लोगों ने लिए, जिसमें आपके सहयोग के माध्यम से, इसी संस्थान के सहयोग के माध्यम से 75 लोगों का सैंपलिंग की गई, जिसमें से 10 पॉजिटिव पाए गए। अभी हमारा अभियान तेज चल रहा है, फिर आगे डेट निश्चित हो गई है। मैं चाहूंगा उस कार्य को जो अभी बात हो रही थी कि सिकल सेल एनीमिया को हम 8 जिलों में, तो हम 4-5 गांव में हमारा साथ आप दे रहे हैं। हम चाहते हैं कि उन 5 गांवों को हम शीघ्र उन्मूलन करें, वो वंशानुगत जो रोग चला रहा है उसको हम कहीं न कहीं तोड़ें, उस कड़ी को खत्म करें और फिर आपके ही नेतृत्व में महामहिम को बुलाएंगे और फिर कहेंगे हमारे ICMR के वैज्ञानिकों ने इन 5 गांवों को जो मॉडल है पूरे मध्य प्रदेश के लिए, आज हमें संकल्प ले। मैं अपेक्षा करता हूं अपने इस परिवार के मुखिया से और आप लोगों से कि हम मिल करके, हमारी डेट निश्चित हो गई क्योंकि हमें समय कम है, काम बहुत करना है, क्योंकि काल करे सो आज कर, आज करे सो, तो वो तो कभी आता ही नहीं। वो तो अपने लोग सब टारगेट निश्चित कर चुके हैं और हर दिशा में कार्य कर रहे हैं, क्योंकि सामाजिक दायित्वों का बोध कराना ही शिक्षा और शिक्षालय और विद्यालय और संस्थानों का दायित्व है। और उसी में आनंद है, उसी में आप मुस्कुराहट पाते हैं। कभी ऐसे नहीं किसी को सता करके, परहित सरिस धर्म नहीं पाई, पर पीड़ा सम नहीं अधमाई। दूसरे को कष्ट दीजिए, इससे बड़ा अधर्म है ही नहीं। दूसरे का हित करिए, दूसरे कोई बीमार है आप स्वस्थ कर दीजिए, आपको मुस्कुराहट अपने आप आ जाएगी उसको मुस्कुराता देख करके। इसलिए मैं आज इस स्थापना दिवस पर हमारे आदरणीय सम्माननीय डॉक्टर दास साहब जिन्होंने हमें आमंत्रित किया, मैं बहुत ही प्रसन्न हूं कि ये मेरा अपना संस्थान है, आप सब हमारे भाई बहन हैं। बहुत बहुत शुभकामनाएं, संस्थान को ऊंचाई प्राप्त करें और पूरे देश में, नहीं विश्व में एक इस संस्थान के नाम से जैसे लोग जाने जाते हैं, जाने जाए। रानी दुर्गावती विश्वविद्यालय परिवार की तरफ से मैं पुनः शुभकामनाएं प्रेषित करता हूं आप सबको। जय हिंद, जय। बहुत बहुत
थैंक यू सर। और अब हम देखते हैं कि ये प्रेक्षागृह काफी ऊर्जा से भरपूर हो गया है सर के स्पीच के बाद। तो एक बार पुनः करतल ध्वनि से हमारे मुख्य अतिथि महोदय के अभिनंदन करें। जो जीवन जीने की कला और मुस्कुराते रहने की जो हमें शिक्षा दी है, उसके लिए हम उनके सदा ही आभारी रहेंगे। मैं NIRTH स्टाफ की तरफ समस्त स्टाफ की तरफ से आपका एक बार पुनः आभार व्यक्त करती हूँ कि आपने अपनी व्यस्तता में से अपना बहुमूल्य समय हमें देकर आज के इस कार्यक्रम की शोभा बढ़ाई और हमारा मार्गदर्शन किया। मुझे पूर्ण विश्वास है कि हमारे शोध छात्र एवं वैज्ञानिक अवश्य ही इससे लाभ प्राप्त करेंगे। अब मैं आदरणीय निदेशक महोदय जी से अनुरोध करती हूँ कि वे आज के हमारे मुख्य अतिथि माननीय प्रोफेसर कपिल देव मिश्र, कुलपति रानी दुर्गावती विश्वविद्यालय को स्मृति चिन्ह भेंट करके हमारी कृतज्ञता एवं आभार व्यक्त करें।
वेलकम, आपकी चर्चा हो।
आपका ही इंतजार था।
आगे आ जाइए, पूरा परिवार है।
तो अभिनंदन समारोह में जो एक चरण हमसे छूट गया था, इसके लिए अभी हमारे समक्ष श्री सुभाष बोले जी, प्रिंसिपल टेक्निकल ऑफिसर, जो कि आज सेवानिवृत्त हो गए हैं और जिन्होंने 36 वर्ष से भी अधिक समय तक अपनी सेवाएं इस संस्थान को प्रदान की। और ICMR की परंपरा का निर्वहन करते हुए मैं मुख्य अतिथि महोदय से निवेदन करती हूँ कि वे श्री बोले जी का अभिनंदन करें। सर प्लीज। आपका बहुत बहुत धन्यवाद।
इस संस्थान में आपने 36
वर्ष 5 महीने
आगे पीछे
और बोले जी का उनके समस्त परिवार जनों के साथ एक बार फिर हम बहुत बहुत हार्दिक अभिनंदन करते हैं और उनके अच्छे स्वास्थ्य की कामना करते हैं। So there is always an end to every event, such as this auspicious event. We take pledge to work earnestly for the growth and development of the Institute from all aspects. We are glad that you joined us to celebrate 39th Foundation Day of NIRTH, Jabalpur. मंजिलें उन्हीं को मिलती हैं जिनके सपनों में जान होती है, पंखों से कुछ नहीं होता, हौसलों से उड़ान होती है. With these words, now I invite Dr. KB Saha, Senior Scientist, ICMR-NITH, for the vote of thanks. So,
good evening, everybody. Today we are celebrating our 39th Foundation Day. And as usual, following the tradition, we celebrate the day with joy and enthusiasm. However, it is our common realization that this day is not only meant for only enjoyment for us, but it is the day during which we fill ourselves with new determination and strength to take up greater responsibility to do productive research meant for the welfare of the marginalized population and meet the challenges as new opportunities in the time to come. So today, in this day of celebration and joy, we are very happy to have with us Professor Kapil Dev Mishra Sir, Honorable Vice Chancellor, Rani Durgavati Vishwavidyalaya, Jabalpur. And in spite of his busy schedule, he accepted our invitation and motivated us with his words of wisdom. I thank you, sir, for joining us here. Sir, your presence not only stimulated us in celebrating our Foundation Day, but it will also strengthen the age-old partnership between Rani Durgavati Vishwavidyalaya and ICMR-NITH. Further, it will promote resourceful and meaningful exchange of scientists, faculties, and students between the two organizations through recently signed MOU, and I believe it will create excellence in academics and research in this region of Sanskar Dhani. We are blessed with your guidance and seek your support in the days to come. Thank you, sir, once again for joining us here. I take this privilege with pride to mention that ICMR has become a household name in India during this pandemic. During this period, when the entire world is facing the terror of the COVID-19 pandemic, the ICMR remains in the front line of the battle in India. We know how tough is the situation and how the policymakers in ICMR are coping up with the situation. Dr. Samiran Panda Sir, Additional Director General, along with the Director General ICMR at the Apex, is steering the council during this difficult time and helping the India's battle against this infection. In spite of his extremely busy schedule, he made it convenient to address our Foundation Day program with his thought-provoking words, as he always does. Sir, we are inspired by your words and seek your guidance and support as well as in our activities. I thank you, sir, for joining our program today. I also take this privilege to thank Professor Hilak for delivering the Foundation Day lecture. It is indeed a lecture which sensitizes us with the nitty-gritty of leprosy and its diagnostics and about the TB, and is very meaningful for us because of the marginalized population with which we dealt with almost every day is vulnerable to these diseases. Looking forward for many more such interactions in future. I thank you again, Professor Hilak, for finding time and accepting our invitation in spite of your hectic commitments. I also thank our director, Dr. Apruv Das Sir, for all his effort, interest, guidance, and support, which helped the Institute in successfully organizing this program. I also thank all the invitees, both in person and those who joined through VC, for making time and valuable presence with us in celebrating this occasion. I also thank all the staff and students who were facilitated today. Last but not the least, I thank all the scientists,
official staff, students who directly, indirectly help the institute in organizing and successfully completion of this program. Thank you, everybody. Jai Hind.
Thank you very much, sir. जैसा कि आप सभी जानते हैं कि सरकारी संस्थानों में आयोजित कार्यक्रमों का समापन हम राष्ट्रगान के साथ ही करते हैं। अतः आप सभी से अनुरोध है कि राष्ट्रगान के सम्मान में अपने अपने स्थानों पर सावधान की मुद्रा में खड़े हो जाएं।
Right.
पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे तब जय गाथा जन गण मंगलदायक जय हे भारत वाक्य विधाता जय हे जय हे जय हे जय 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 हे
भारत माता की जय भारत माता की जय भारत माता की जय
जय हिंद, जय भारत। I would like to invite you all to join us for dinner served outside. And thank you once again for joining us and making this event memorable. Have a wonderful evening.